In this video, we are going to talk about the Oncovirinae. These are basically RNA retroviruses, so that's the first thing to keep in mind. So they have an RNA genome and they're retroviruses, right? Which means they're not going in the same direction that we expect for replication. Both are associated with the development of cancer, and they're all in the Oncovirinae family. They were originally called basically just RNA tumor viruses. They're known to cause leukemias, sarcomas, and lymphomas in many animals. They're non-cytolytic, and that's actually really important. Their main mechanism is cell transformation and immortalization, and that's what actually leads to the development of disease. In most cases in this video, when I say the word latency, most of the time when I talk about latency, I'm talking about you had an infection, you got sick, you got better, but the virus hung around and later caused a different disease. So like in varicella, when you had chickenpox and then years later you get shingles, or herpes, you have a cold sore, years later you have another cold sore. This is different. You got infected. You maybe felt something, maybe didn't really matter. But what does matter is 30 years later you got cancer, and it's all because you contracted HTLV1 at some point in your life. So what are they? So there are actually a lot of different Oncovirinae, or HTLV1, which literally stands for human T-cell lymphotropic virus. But there are only 3 that are known to cause disease in humans: HTLV1, HTLV2, and HTLV5. Only HTLV1 has been definitively associated with disease. HTLV2 and 5, it's just kind of a rumor. HTLV1 is able to replicate but cannot transform cells in vitro. What does that mean? It's really hard to study. We can't really culture it in the lab and see the exact same result that we would see in a human. HTLV1 also has an incredibly long latency period, greater than 30 years, 30 to 50 years from the time of infection. The thing it's most known for doing is causing ATLL, or adult acute T-cell lymphocytic leukemia. It also is associated with myelopathy, which is tropical spastic paraparesis. It's a non-oncogenic neurologic disease that's caused by HTLV1, but the one I most want you to think of is ATLL. HTLV2 has been associated with forms of hairy cell leukemia, but it's not a definitive connection. And HTLV5 has been associated with malignant cutaneous lymphoma, but again, not a definitive connection. The only one that we really can definitively say has a role is HTLV1. Okay, so what is it? How do we get it? First off, HTLV1 is spread in fluids: blood, sexual transmission, and breastfeeding, which is actually really important, as you'll see in a minute, for kind of the epidemiology. Basically, it enters the bloodstream, and much like HIV, it's going to infect your CD4 positive helper T cells, all right? But remember, in this case, unlike HIV, it's not killing them, it's hiding in them. It'll then stay in those for a long time. So in addition to blood and lymphatic organs, these T cells are also going to reside in the skin, which is going to contribute to some of the skin lesions that we see with ATLL. Neurons also express a receptor for HTLV1, so that's another place you might see it. So basically, you get infected, and you can get cell-to-cell infection between your CD4 cells. You get a carrier state for a long period, 30 to 50, up to 60 years, okay? At that point, you have oncogenesis of the infected cell, and this oncogenics is actually due to the TAX protein. TAX protein is basically a transactivator, and what it does is it targets the IL-2 receptor, or IL-2. What is IL-2 again? IL-2 is T-cell food, right? Anytime we have a lot of IL-2, I want you to think of your T cells just proliferating like crazy, and that's what happens. That's why we develop this leukemia, because we suddenly get mass proliferation of our T cells. Okay, so who gets it and where? Like I said, it's transmitted along the same routes as HIV, and it infects about 5 to 10 million people worldwide. So not a huge, pandemic, but still a lot of people. It is endemic in certain areas, particularly southern Japan, the Caribbean, Central Africa. You can see there's a bit of a increase there. And among African Americans in the Southeast United States, we also see it in Central America, so kind of this equatorial distribution with some other areas. In Japan, it's actually kind of interesting. There are certain regions in Japan where we actually think incidence is upwards of 35 to 50%, but it's really hard to get a good estimation of what the worldwide kind of viral load is, or how many people in the world are actually infected, because there isn't really standardized testing across the board internationally. We worry about it a bit in the United States, not very much, but basically where you worry about it is IV drug users and the HIV population. The HIV population, because obviously that could be very dangerous. IV drug users, because there's increased risk with any bloodborne pathogen there. In the United States, we actually test both blood and organ donors for HTLV1, and that actually helps limit the spread. The other thing we really worry about is breastfeeding. So in certain areas, like in southern Japan, where it's 35% potentially, a lot of infants actually contract it during breastfeeding from, their mothers who potentially contracted it either when they themselves were young or from sexual intercourse. Okay, so what's the syndrome? It's usually asymptomatic, but it can progress to ATLL in about 1 in 20 people. So pretty high odds if you contract it. 30 to 60 years. It's basically an acute or chronic neoplasia of CD4 positive T cells. And the malignant cells are often termed flower cells, because look at these beautiful flowers. They have these beautiful petals, because basically what happens is it's a lobulated nuclei, but it really is beautiful on a blood smear. Patients will typically exhibit an elevated white blood cell count and skin lesions similar to those seen in the Sézary syndrome. Unfortunately, ATLL is usually fatal, even if we treat. It can also cause other diseases, like I mentioned, including uveitis, infectious dermatitis, and other inflammatory disorders. So how do we diagnose it? Now, if you contract HTLV1 and you don't have cancer yet, theoretically we could find it using ELISAs for either antiviral antibodies or virus-specific antigens in the blood, and RT-PCR because it's a retrovirus for viral RNA. Here's the rub though. There's currently no approved treatment. Some people have used AZT, a drug that we use for HIV, with interferon alpha, and that may have some success, but there's no approved therapy. So currently our best bet is to prevent. So sexual precautions, screening of blood and organ supplies, and increased awareness. Although all of that said, it's very difficult to stop vertical transmission from mother to baby.